the first seminar of this school year for the CFAR Substance Use Program of Research. And myself and Judy Hahn and Carol Dawson Rose, as well as Jennifer Jane, are part of the UCSF CFAR leadership coordination group with Yvette Kuka. And one of our goals is to increase collaboration and information sharing on folks who are interested in the intersection of substance use and HIV research. And Adam Carrico is our seminar speaker for today. And I am pleased to introduce Adam, who is a licensed clinical health psychologist with expertise in developing and testing biobehavioral interventions to optimize HIV prevention with sexual minority men, or SMM, who use stimulants such as methamphetamine. Adam's clinical research program focuses on boosting the effectiveness of contingency management for HIV prevention, and he also conducts translational research that focuses on elucidating the epigenomic and microbiome gut-brain axis pathways that underlie the neuropsychiatric comorbidities in people living with HIV. And today, Adam is going to be sharing his projects. And Adam, did you have a preference for questions like during or holding
Oh,
it off
please
at the end?
interrupt me. I'm happy to, talk about things as we go. I think that's much more fun actually than me just talking at you.
Sounds great. Well, without further delay, everyone, please give Adam a warm welcome.
Yeah. Nice to see you all, many old faces, some new that I've met and work with at UCSF that I didn't know when I was there. So really cool. It's been, I think, about we're going on 9 years since I moved, which is, I'm not sure where time
Wow.
goes either. Um, but so I titled this talk Parts Unknown. It's my homage to Anthony Bourdain, who we all know and love. And, it's really at for me, it's the unknown part is this intersection of behavior and biology and some kind of just thoughts on how I came to do this work. I tell people, here's how you become a weirdo like me, right? That's crossing these boundaries and working across disciplines and often doing things that I shouldn't do as a psychologist. So, I hope you walk away from this talk realizing that a PhD is really like a library card or an MD for those of you that are students. And, you get to decide what you want to study and learn about as you move forward, which is really kind of exciting. And you see the right slide presentation view, yeah? Good. So my, kind of training as a doctoral student was in this field of psychoneuroimmunology, which looks at mind-body interactions and specifically kind of neuroimmune signaling. So how stress, depression, other psychological experiences impact neuroendocrine hormone regulation. So cortisol, norepinephrine, and how that in turn impacts immune function. A lot of the work in psychoneuroimmunology was done, or PNI was done prior to the heart era. So it's much older. It's work showing that depression, for example, predicts faster HIV disease progression, which is a enduring finding. Also PTSD and HIV disease progression. More in the modern era, it's been harder to establish those relationships. When we have such a great ART regimen that everyone's taking. Uh, so, so that's kind of where we're at with P&I research, right? That it kind of died out. This mind to body stuff is interesting, although as I was walking away from my graduate training, felt like maybe it's not as clinically meaningful. And there are two kind of choice points in my career that have directed me to what I do. One was really around methamphetamine use. The early trials that I worked on in PNI, my primary job was to exclude all the people who use substances. So I got really good at like building rapport, doing a structured clinical interview, getting people's trust, and then promptly excluding them from the trial. So I had, as you can imagine, a lot of really provocative clinical interactions with people that use drugs as to like, Why am I not in this study? Why are you discriminating against me? Why can't I be part of this? And I'd have to explain the inclusion criteria and try and find them another study that was a good match. But I came to wonder about methamphetamine use and just substance use disorders broadly as prevalent comorbidities in people with HIV that complicate disease management for sure, but also could be exacerbated by HIV pathogenesis. So that kind of bidirectional causality. The other main choice point or kind of fork in the road came when I was in Miami. I got really excited by the gut microbiome. And of course, in HIV, we know that HIV alone does a lot of damage to the gut very early in infection, like within the first 14 days. And that is associated with altered gut microbiota, although that's a bit controversial still. We're not entirely sure if it's HIV or if it's receptive anal sex that's leading to that altered gut microbiota. But we know that stimulants like methamphetamine also have potent physiologic effects. They raise sympathetic nervous system activation, heart rate, blood pressure. We blow out norepinephrine at our nerve terminals for the sympathetic nervous system, and that directly enhances HIV replication. In the gut, we know that stimulants like methamphetamine accelerate or potentiate gut damage, and bowel motility is kind of enhanced, right? So there are lots of ways in which they alter the functioning or the physiology of the gut that could make the microbial products leak out a bit more effectively. So this process of microbial translocation is when bugs that should stay in the gut kind of make it out into the periphery. Things like lipopolysaccharide, which is a classic bacteria. And once they're out in circulation, the body recognizes them as invaders, right? It says this bacteria shouldn't be there. We've got to mobilize, we've got to kill it, we've got to neutralize it. So you get an activated immune system. One of the major ones that we study, hang on. One of the major ones that we study is soluble CD14. So when monocytes are stimulated by LPS, that's a mechanism of release of these soluble CD14 molecules or targets. So immune activation happens. Concomitant with that, there's more inflammation. Your immune cells are generating inflammation to neutralize pathogens. So there's this way that the co-occurrence of HIV and meth use could accentuate or exacerbate the underlying HIV pathophysiology. And coincidentally, many of these changes to the gut, the leaky gut, and the immune system persist even when people have suppressive antiretroviral therapy. So we have persistent immune activation and inflammation in people with HIV that partially stems from this leaky gut as well as the HIV reservoir. But so once your immune system is angry and activated and inflamed, there are lots of ways that that directly impacts neurobehavioral function. Activated monocytes, inflammatory cytokines like IL-6 can cross the blood-brain barrier. And the blood-brain barrier is preferentially leaky. So there's a lot of evidence that the prefrontal cortex is particularly vulnerable. And when the inflammatory cytokines and activated monocytes make it into the brain, they induce kind of immunometabolic dysregulation that enhances neuroinflammation and depletes dopamine. So it's this kind of endogenous anhedonia that we see in people with HIV. We know, for example, that the prevalence of depression is about 3 times greater in people with HIV. And part of that is due to stigma and all the terrible things that people with HIV endure. And it's partially due to biology, right? Some of this persistent inflammation damaging the brain. So there are these direct pathways here. A lot of our work is focused on amino acid precursors for neurotransmitters. So serotonin has precursors. It's called, oh my goodness, I'm going to forget it. Tryptophan. And so we look at the tryptophan kynurenine ratio as a marker of whether or not someone is degrading their building blocks for these. And then there's the phenylalanine tyrosine ratio for dopamine. So we think that robbing these building blocks of neurotransmitters partially induces neuropsychiatric symptoms like depression, anhedonia, impulsivity. We have some data that there's an association between the behavioral analog risk task, which is a balloon pumping task of risk taking, because you pump the balloon until it pops and you make more money as you pump. So you have to decide when to give up, right? And are you going to gamble? It's a fun task that our participants like, but we see that those that have depleted tryptophan in their blood had more impulsivity on the BART, for example. So there's, I think, lots of cool work being done in the field to understand how these HIV-associated gut immune dysfunction parameters impact neuropsychiatric functioning and the extent to which they're amplified even more by stimulant use is of great interest to us and others. So when there's this endogenous gut immune dysfunction that creates neurobehavioral alterations like anhedonia, what happens is it's a positive feedback loop. People are deprived of pleasure and reward, and they hit the lever for more meth, right? Which is an endogenous driver of dopamine and a potent kind of activator of dopamine. And so, a lot of our work has been trying to understand some of these neurobiological pathways in the hopes that we could inform interventions for people with HIV in particular, and those that use methamphetamine specifically. Any thoughts on this or questions? I kind of went through a lot of stuff.
Um, Adam, you mentioned that you're mostly referring to methamphetamine. Some of this also for other stimulants like cocaine and like MDMA.
Yeah, it's I always get dinged on this too in grants, just so folks know. I think of stimulants broadly as a category or a class of substances. And I think about indirect effects, right? Through the autonomic nervous system, through the hypothalamic pituitary adrenal axis, the gut-brain axis. But there are direct receptors for different drugs of abuse on immune cells, right? And for dopamine on immune cells. And so, there could be heterogeneity in the outcomes. I would say also that methamphetamine has a much slower half-life than things like cocaine. And so it's a potent and durable physiologic stimulator, whereas cocaine is much more stochastic, right? Depending on how people are using. So there's a lot of nuance there, right? But when we work with actual patients, it's like polysubstance use is the rule, not the exception. And so I think a lot of those distinctions are kind of academic. But it's the benefit of us to kind of like pair with basic scientists because they can bring immune cells into the laboratory, they can cook them up with meth, with coke, with these other drugs of abuse, and have a much more controlled environment to study its immune effects. Yeah.
If it's not too much, I have one other question, Adam, which is do you see a dose response to the inflammatory consequences of stimulant use? Like if there is reduction, is there a reduction or vice versa?
Yeah, it's so a lot of our work early on, we have a trial called Artemis that I'll talk about that you may know. And so that was among people that use methamphetamine. And so we saw dose response associations of recent use, which was also linked to more frequent use, right? So people that use meth more frequently had elevations in a lot of these different parameters. We also see routes of administration associations. So particularly men that were injecting methamphetamine had a much more robust profile of elevations across all of these parameters. It's unclear if that's due to the physiologic effects of meth or if it's the route of administration causing things like endocarditis or abscesses or infections that are driving some of these symptoms or some of these perturbations, rather. So it's kind of wild, but the dose response piece I think is really critical, especially around causal inference. I think it gives us a much greater clue, especially in observational data, if we can see those dose response associations. So we're often in studies now going into people's urine samples that are reactive and quantifying their metabolites for methamphetamine and other stimulants so we can look at some of those dose response pieces. We see that with, I think, troponin, actually, Elise. a dose-response association of meth metabolites with troponin, but it's in like 7 or 8 people, you know.
Sorry, we were looking at cocaine, but there was definitely a dose response. Yes.
Yeah. And we did it with meth and found one in some data that Michaela's working on my team. So, you know, it's nice when we're actually looking at the dose data. Right on.
I just want to add that that was like the best summary of that whole picture that I've ever heard. And I'm going to
Oh,
go back
good.
and listen to you say it again because the was the part that you said about the brain, that's the part that sounded sort of new to me. Is that like really, it sounded like almost like leaky brain. Can you say that again, maybe? I
Oh,
got
yeah,
a little
totally.
confused.
And so there's the article, I can send it out for the group. It's by Lucido, Lucido. And so he works with the Emory folks, you know, like Jen Felcher and others that are really into the PNI world these days. And so we know in across the general population, this is not specific to HIV, that the blood-brain barrier is preferentially leaky and it receives input from the periphery, from the body. So like these inflammatory signals are
Okay.
crossing at specific areas in the brain, like the prefrontal cortex, and I think at the nucleus accumbens, which is critical for our reward processes. And so once they cross in, they're potentiating neuroinflammation, they're changing the immunometabolic rate of cells, and depleting things like dopamine, increasing, you know, other pieces that would lead to neuronal death. And so it's a fascinating literature. And we know that in HIV, because there is so much systemic inflammation, that these pathways could be even more relevant.
Thanks.
I have a good slide on it too that I'll send you from the group. They have excellent pictures in the review.
Can I ask you a quick question about just baseline gut damage and microbiome? Because I imagine that even early on in infancy and even as fetuses, you know, we start to develop our microbiome and there are differences between individuals. How do you measure that, or have you measured that in your studies?
It's so interesting. And we have different microbiomes across regions of the world, right? Based on diet, based on, you know, just a variety of factors. And so in our work, because we do it in the context of clinical trials, we're able to kind of look at changes secondary to intervention effects. So that helps us kind of disentangle, you know, kind of someone's baseline state from how we're able to change it. But you know, this whole idea of gut dysbiosis that people talk about, or this dysregulation of the microbiome, we're not entirely sure what that means, right? There's no clinical kind of composition of the microbiome that's pathogenic, for example, but there are bugs that we know are more inflammatory on average. And we see those in sexual minority men that engage in receptive anal sex and others. So the microbiome is a fun area. I Sometimes it's very difficult, though, to observe change. There are a whole bunch of, for example, like probiotic studies in macaques, in people living with HIV, and they're really not able to shift the microbiome that much. So, you know, there are the bugs, and then what they make, the metabolites. And so I think the current thinking is that maybe the metabolites are more relevant, and maybe we could do supplementation with metabolites even when someone's microbiome is going to remain stable or off, you know. Um, it's there's so much to be done there. Yeah. And we do have those samples. We get poop samples now, which I'm really excited about, on a trial called Trident that I won't talk about, but it's a depression treatment trial where we're trying to shift the gut-brain axis. So we look at poop samples from microbiome and its metabolites, immune cell signaling in the periphery, and repeat fMRIs. So we're hoping that those data will help us kind of put the pieces together. Yeah. Neat. And so, you know, we have done a series of studies over the years with Anissa and others, and Brad Oizerot, who was at UCSF when I was there as well. It's just been so fun to work with both of them. And so I refer to these as outside-in pathways. So things that people are doing or think being done to them that impact their immune function. So in this instance, we looked at a dose response kind of association of recent stimulant use. So these were individuals that were all virally suppressed. We had 55 of them. And those that had a stimulant tox positive urine, 9 times out of 10, this was methamphetamine, maybe 9.5. And then occasionally cocaine was in the mix too, but maybe 1 or 2 cocaine only. And so we found that those with the tox positive urine had 32 differentially expressed genes for HIV latency, which was fascinating, cell cycle regulation, and immune activation. When you do this epigenomic work, it's just as much about how pathways or families of genes change rather than a single gene, because we know they act in concert. So we looked at 2 directional perturbation, so ways in which pathways were getting pushed in one or pulled in one direction and then pushed in another, right? So this kind of fighting back for homeostasis that the body does, that's a chronic kind of stressor for the system. And we saw a variety of pathways that were exciting to us. NK cytotoxicity for me as a PNI person, that's a common outcome in our work. T cell receptor signaling. And of course, I was so excited to see tryptophan and tyrosine metabolism being altered in recent users. So this gave us some indication that HIV could, or methamphetamine and other stimulants maybe, could be inducing HIV latency physiologically, and altering a variety of pathways relevant to immune function and neurotransmitter synthesis. We kind of moved on from these a bit. One prominent problem or critique with the Artemis data was everyone was living with HIV. So we weren't really able to look at the interaction of HIV with methamphetamine versus without HIV. And so we did the full 2x2 here in a very small COVID recruited sample of sexual minority men in Miami. And after we controlled for HIV status, those who engaged in stimulant use had higher soluble CD14. This replicated a finding we found among meth-using men with HIV in 2018. This was led by Emily Shirinak, who is a postdoc at University of Miami. And you can kind of see here that the association is the same irrespective of HIV status, right? Maybe it's a little stronger in the negatives, but that didn't seem to pan out statistically. So where people are using stimulants with the triangles here, this is their predicted probability of a CRP 1 or greater. So stimulant users had elevated CRP as well as a higher mean soluble CD14. There were no effects of SARS-CoV-2 infection, either RNA positive or nucleocapsid antibody positive on these pathways, which I think is fascinating, although a small sample and difficult to determine. But a good direction for future research to understand these. Emily has gone on to do a lot more omic work. So she was able to take the plasma from 61 of these participants with and without HIV. We conducted targeted metabolic profiling using Biocrates. We were able to look at 390 metabolites, so this becomes big data. And so she looked here, this is, I think, a wonderful figure, just around what specific metabolites were up or down regulated after we adjusted for the false discovery rate, or the FDR. And in general, the pattern that emerged was, again, that the stimulant use and HIV group, those with both, had greater immune activation, metabolites relevant to addiction, actually, and cardiovascular disease. We're seeing some CVD risk in other work that's being done with like troponin, as I mentioned. So that's of great interest to us. And I think more broadly, we need better interventions to reduce risk of CVD in people with HIV, especially those that use substances. And we're kind of hot on the trail of some of that in upcoming grant. And another piece I think that's kind of opened my eyes is, being in public health for quite a while now as a psychologist, we start to see the role of social determinants of health. And I have to wonder, are these really the physiologic effects of substance use or minority stress or other exposures, which we call the exposome? Developmental adversity counts in this, right? Are they the physiologic direct effects of those exposures, or are they due to some, kind of multi-level effect modifiers, if you will? There could be indirect associations of things like neighborhood characteristics on substance use and then to HIV pathogenesis. Multi-level structural and neighborhood factors could moderate those associations. So maybe we see substance use as worse if you live in a good neighborhood. Because if you live in a bad neighborhood, everyone is having worse function irrespective of that. And there could also be direct associations of these multi-level drivers or social determinants of health with HIV pathogenesis. And so, I think we need to increasingly contextualize substance use. And to me, this destigmatizes it and helps to understand the complexities of the lives that our participants have without kind of focusing only on the substance use. A lot of this work has been led by Delaram Ganouni, a former PhD student who's now a junior faculty with us. She's been very passionate about looking at structural and social determinants in our data. This is from Delaram's dissertation, more of the outside-in piece, but she found structural factors were associated, or like unemployment, homelessness, and history of arrest were associated with immune activation, the soluble CD14, and inflammation, the TNF-alpha R1. This SCD163 is another monocyte macrophage activation marker. And you see on the right here some of the box plots around homelessness and arrest with soluble CD14. So, it's, I think, exciting to think about the complexities here and what these might be proxies for. Especially history of arrest is such a potent barrier to employment that these are really prominent determinants. Right. Delaram's also gone on to look at neighborhood level characteristics. So we were able to kind of use the data from Artemis participants and extract their neighborhood information. So you need their full address and you geocode it and you get, down to the neighborhood level. So she looked at the association of neighborhood level adversity. We chose a few indicators, percent below, 150% below the federal poverty limit, percent unemployment in your neighborhood, percent without insurance, and percent racial ethnic minorities in a neighborhood. This one became particularly controversial among our group on how to interpret and especially how to frame. We view this as an indicator of, redlining, structural racism, many of the factors that have led to disenfranchised, impoverished, predominant minority neighborhoods. And this was all done in San Francisco. So you think about Hunters Point, for example, and just the structural stigma and racism that has led to segregation in our society. So she looked at these neighborhood level determinants and their direct associations with inflammation, immune activation, and cellular aging. For cellular aging, we're able to look at people's DNA, their actual extracted DNA, and you measure the protective end caps on their chromosomes. So it's kind of like a shoelace that keeps it from unwinding. And so that's what your telomeres do. And as they shorten, it's a measure of biological aging. And so we thought there would be direct associations and indirectly via stimulant use. We found that neighborhood level adversity was associated with higher inflammation, IL-6 and TNF-alpha. And although there were no direct associations of neighborhood level factors, we did find they were associated with stimulant use. We also found that stimulant use was associated with shorter telomeres. So a nice, again, cross-sectional study, 110 sexual minority men with HIV that use meth, but a lot of more work to do here. This work is in Press with Health Psychology. There's a special issue on SGM health. I think Anissa has one in Press too, which is fun. So we're excited for that. Yeah. Oh, and coincidentally, we got challenged by reviewers to adjust for someone's individual level SES. And I was like, oh my God, this is terrible. Like, it's 110 people. What are they asking for? It held up. So lucky us, I guess. But so it's not just that your individual SES is your neighborhood SES is a proxy for your own SES. It's the neighborhood effect over and above the individual, which was a lot of fun. Oh, I did have this slide. So this was a slide I was thinking of, Judy, where it's, talking about the inside out pathways, right? How these HIV-associated pathophysiologic alterations in the periphery and in the CNS could damage the brain. So HIV and inflammation are associated with a variety of psychiatric comorbidities. I mentioned that inflammation amplifies catabolism of tryptophan, which is a building block for serotonin. Inflammation also preferentially alters frontal limbic structures relevant to reward and dopamine. So this is your dopamine reward system here. The VTA and the SN, they project into the ventromedial prefrontal cortex and limbic system structures. So there's a lot of connectivity there between those two areas where inflammation is leaking in. And it's much to our adaptive benefit, right? If you have an infection, if you have some disease that could be transmitted to your community, we have this built-in pathway to induce sickness behavior where people become withdrawn, they're not engaging. And so, we think this evolved naturally in our species because these individuals were able to avert infection of a herd of people with a lot of uh, the same virus. So here in the periphery, we know that inflammation upregulates the activity of the immune cells, so the NF-kappa B, the metabolic rate. It also induces glutamate and reactive oxygen species, so you're damaging mitochondrial function. You're increasing glycolytic metabolism, so the use of glucose. So it's just kind of revving everything up and creating, a lot of pieces in here, oxidative stress that could damage the DNA, that could damage the mitochondria and their function. And then when these cytokines cross over, as you see here, it increases glutamate, which is associated with cell death, neuronal death, and decreases dopamine. So, this overall leads to that kind of presentation that we see across many psychiatric disorders called anhedonia, where it's really the lack of pleasure in normally rewarding activities, decreased effort expenditure, and just decreased reward from even primary reinforcers like food, money. And coincidentally, we see this in people that use substances. We put them in scanners with peers that don't, and we see that they just have less robust activation of that dopamine reward system in response to even primary reinforcers like food, money, or sex. So this anhedonia is a cross-cutting symptom. We often think of it as relevant mostly to depression, but increasingly we see it as a prominent feature of substance use disorders. Any thoughts on this before I go on? And here's the Lucido reference that I was talking about. It's super cool. I have it on my desk to read. Do you know what I mean? This is one that it's like that long where you kind of have to dig into it. So maybe over the Christmas holidays, I'll get there. So there's another inside-out pathway that we've been looking at in the RADAR cohort with Brian Mustansky. So I was really fortunate to be a consultant on RADAR and just have access to this impressive cohort of 1,300 sexual and gender minority people. Brian is particularly focused on young people, so they were recruited from age 16 to 29. They were followed for a median of 5 years, which is amazing. Youth with HIV were mostly enrolled at baseline, 195, but there were 85 seroconversions over follow-up. And we were able to look at existing data in RADAR on systemic markers of inflammation. So they measured C-reactive protein, or CRP, TNF-alpha, and the third one will come to me, IL-6. So we looked at those three pro-inflammatory cytokines in the subset of 834. We looked at Cox proportional hazard regressions, trying to understand meth use as an infectious disease. So we looked at incident first-time methamphetamine use. So this was the first time in someone's life that they had reported using methamphetamine, because we got lifetime self-reported use at baseline. So it's a really cool kind of outcome and something that we can only look at in young cohorts. So lots of good reasons to work with young people around the HIV epidemic, of course, where the majority of transmissions are happening. But we can understand these initiation of these using these substances and try and prevent that, hopefully. So we had a variety of measures in RADAR. We controlled for housing instability, HIV status, and so on and so forth. Substance use, we had cannabis, cocaine, non-prescribed use of stimulants, depressants, or opioids. We had to like combine prescription drug use because there wasn't a bunch of it. We looked at meth prevalence by year. So seeing, is there any evidence of increasing meth use, and would that vary by age? We did see increases, predominantly in those that were older. So the median age of methamphetamine use was around 23. So, people are starting this post-college, early adulthood, right? And it's an interesting developmental period to think about interventions. And again, we looked at incident first-time methamphetamine use via self-report or urine toxicology. So we're able to somewhat mitigate underreporting. And this was the, I think, take-home of the paper, that we found independent associations of prior HIV diagnosis and C-reactive protein with increased rates of first-time methamphetamine use. So youth or people with HIV had a twofold greater rate of starting methamphetamine after their diagnosis. So we often think of meth risk leading to risk for HIV, but this is the opposite direction, where people that had not reported using meth in their lifetime and their HIV diagnosis preceded initiation of meth. I think there are tons of reasons for this. One of them being that our prevention campaigns are so focused on HIV. Don't use meth, avoid HIV, right? But we never talk about the harms of meth alone, especially for people living with HIV. And so there just aren't the norms and the education out there, I think, to help people with HIV avoid starting meth and have great reasons to do so. And then elevated systemic inflammation in C-reactive protein, 18% faster rates independent of HIV status. There were no interactions of inflammatory cytokines with substance use, which would have been really cool. But like that this would be more or less pronounced, for example, in cocaine or people with HIV and so on. There's a bit of evidence for gateway drugs here. Cocaine use, threefold greater rates of meth use, any cannabis use, twofold greater. So I think lots of good data here to inform preventive interventions. We know the developmental period. We know the risk exposures. We can identify those youth or those people and try and intervene to prevent initiation. So we're very interested in, or I'm very interested in thinking through or partnering with people that might want to do interventions in this area. Especially our positive affect intervention, I think, is a great choice given the anhedonia that people experience that may drive them to use methamphetamine via this inflammatory signal.
Adam, do you happen to know of other groups that this is fascinating that you're finding that people with HIV then subsequently use cocaine. And we did a study where we found that women experiencing homelessness and did not use stimulants at baseline used them later, which is very similar. It's like, we always think about stimulant use leads to homelessness or stimulant use leads to HIV, but actually there is quite a bit of, it's like, at least in our studies, we're having evidence for the opposite. Do you know of other studies or other groups that are looking at this where we automatically assume that substance use leads to things, but actually these things can lead to substance use? Are you guys or other groups doing more in this area?
I would love to. I'm not really I think that we haven't done enough work overall, and it's a great piece to think through how we could build more momentum in this area because it just challenges some of the, I think, ingrained kind of ideas that we have about causal inference. And it's not negating that pathway, right? Clearly, meth use
Right.
does enhance risk for HIV,
Yeah,
but we have to think about, is the reverse also true?
totally. Yeah.
Yeah, I'd love to do more stuff in this area. We're working with an acute HIV cohort in Thailand, and there are about 40 people of 180 that use methamphetamine at acute HIV infection. By 96 weeks, we have about 15 of those still using meth. So it's a great place. I think, Judy, like your work in Uganda, like how do we study that natural change? Is it real? So we're trying to get urine samples and figure out, is there underreporting going on because they were stored, but then also to study some of these bidirectional connections. The sample size is just pretty small, but we kind of need a cohort of people that use meth with and without HIV, and then we'd be able to answer a lot of these questions. And then, this gut immune dysfunction is really the hallmark of HIV. I talked a lot about this, so I won't really belabor it. This is a gut-brain axis review that we did in psychosomatic medicine, just outlining some of these outside-in pathways and some of these inside-out pathways for the bidirectional communication between the brain, the gut, and the immune system. I'll present some data from the acute HIV cohort. This is an outside-in kind of overview, though. So as I mentioned, 1 in 3 people with HIV are depressed. Depression's consistently been associated with negative outcomes, such as difficulties with ART adherence, elevated viral load, virologic rebound, faster all-cause mortality, even after adjusting for viral load. There's something about depression that is uniquely risky for people with HIV, even after we account for health behaviors that should explain it. So it's still a very interesting phenomenon to address in and of itself as a prominent comorbidity for people with HIV that also still reliably increases risk of progression. And in RV254, this is another inside-out thing. So these folks were enrolled, it was sexual minority men in Bangkok, Thailand. We worked with Rob Paul at University of Missouri St. Louis, and they do a lot of machine learning. So I would think of doing this work with like a latent class analysis or a latent profile analysis. That's kind of the machine learning HDB scan with UMAP. Was how we got these 3 kind of latent trajectories over 96 weeks. So there were those that were persistently elevated in their PHQ-9 score greater than 5. There were remitters, which we found really interesting, and there were those that were consistently low even at diagnosis. So, very interesting groups here that we've tried to study. And so once we identified people's class membership, we're able to look at correlates. And so this is led by Jen Chavez. She's a PhD student in my lab. She just got an R36 to extend this work and do some proteomics with samples from that cohort. So I'm really excited and proud for her. She's very into mitochondrial dysfunction, which has gotten me thinking about it. There's so much fun synergy between students, and it's just such a pleasure sometimes. So in this, what we did, we compared those that were persistently elevated in their depressive symptoms versus those that were low. And we did find some association of substance use, I would say chemsex drug use, so ED meds, poppers, and meth being marginal. And interestingly, we did find that peak viral load was associated with greater risk of persistent depressive symptoms. So the peak viral load was higher, indicating maybe there's more penetration in the CNS, greater reservoir, more inflammation, more damage to the brain, just plausible mechanisms. All right, go back. And then last, we found inflammation was elevated at acute HIV infection for those with persistent depressive symptoms. So much of the same story here, right? Although in this instance, we're able to look at the very earliest stages of HIV infection and see that that matters for people's long-term mental health, even though they've been receiving suppressive ART, right? So what's done during those first days of infection, the first month of infection, is almost a set point for some people. Of course, this is not deterministic. It's, there's a lot of variance there, but it explains some of the trajectories that people experience. All right, I will briefly run through treatments. You all know we have a lot of treatments that are behavioral. Behavioral treatments are first line for methamphetamine use disorders. As you all know, I'm sure we have no approved FDA-approved pharmacotherapy. So there's no buprenorphine or methadone or what have you for methamphetamine use disorders. Really, CBT is kind of the mainstay in contingency management. We've done a lot of work with the Stonewall Project in San Francisco, implementing these from a harm reduction perspective, which is near and dear to my heart and so much fun that we were able to work with them. And our trial was done with their contingency management program. So they've been delivering incentives, tangible incentives, as positive reinforcement for meth abstinence over, the past 20 years, probably. And we know from the literature that this is a highly effective intervention. It achieves moderate reductions in stimulant use, a Cohen's d of 0.66. And there's some evidence that CM alone reduces viral load in the short term. But what we were interested in this trial is how we could take the benefits of CM and extend them after the incentives end. The perception in the literature has been that CM, the incentives, the benefits that people gather rapidly abate after the incentives are done. That's not true, actually. And there's a great meta-analysis out showing that CM actually persists greater than CBT. So that's been fun to see. But the premise of Artemis was before all this. And so we were really interested in extending the benefits of CM. And Judy Moskowitz, I worked with as a co-primary mentor at UCSF. It was just so wonderful to work with her before she went to Northwestern. And we did a lot of work on positive affect and positive affect interventions together. My work has really been to apply her positive affect intervention to the context of substance use disorders. And we think it could serve two important functions. Getting time away from methamphetamine use leads to withdrawal, symptoms of depression, lethargy, anxiety, a lot of bad stuff that people may need help coping with. So providing those positive affect skills and coping skills during contingency management could help people get more negative urine screens or make it through more effectively. I think more importantly, positive affect addresses this underlying anhedonia or hyporesponsivity to reward that we see in substance use disorders. We're really trying to sensitize individuals to natural sources of reward and reinforcement, and hopefully changing the payer mix of their dopamine reward system, right? Positive affect interventions could address these processes that we think maintain a substance use disorder. Judy's prior trial was done in people recently diagnosed with HIV. We found that it reduced HIV-specific distress and use of antidepressants. And so Artemis was the second trial that we did. And so we enrolled 110 sexual minority men with biologically confirmed meth use, not us. At baseline, we surprisingly only had 14% of participants with unsuppressed viral load. This is when the new medications were coming out around 2013. The NSTI meds, you could achieve viral suppression at a much lower level of adherence. So although we were worried seeing all of our baseline results coming in, we extended the follow-up to 15 months in the hope that we would observe differential rates of virologic rebound in the intervention group. And that's exactly the story of this trial. Artemis, the positive affect intervention, the 5 sessions, was able to kind of maintain people at their baseline level of unsuppressed viral load. And then the attention controls, about half of them had at least one unsuppressed viral load over the 15-month follow-up. So again, it's a small trial. It's the precision of these estimates, I wouldn't take them to the bank. We have a number of trials that we're following up with that I can give you some insight into, but very interesting work. We also worked with Brad And so we looked at people and their DNA, and we did arrays to understand the methylome. And so we looked in a subset of individuals that had sustained viral control. So they were undetectable at baseline and undetectable at 6 months. And so we used their DNA only because it's such an expensive test. We didn't have enough to do the full sample. And so we looked over 6 months over the period where we saw self-reported increases in positive emotion, self-reported reductions in stimulant use. We looked at a few target methylation sites for neuroimmune signaling, things like norepinephrine and cortisol, but also oxytocin, which is an affiliative hormone. It induces lactation and labor, so that's where we know it most in medical work. But it increases affiliative behavior and has been shown to alter drug-related reward in rats, which is fascinating, and BDNF, which is kind of a neuroprotective hormone or factor. And so what we found was there was decreased methylation for beta-2 adrenergic receptors on immune cells. So these are the decreased methylation would lead to greater expression because you're opening up that gene, it's getting coded for, and the receptors are populating. So with declining meth use, maybe the body's recovering and saying, okay, we can actually have more receptors for norepinephrine and other hormones that are induced by meth. And we saw that oxytocin receptors were also demethylated, so potentially greater oxytocin receptor expression, which has been so fun for us. I could talk about oxytocin for like a year. We proposed a trial on intranasal oxytocin, which people totally hate, so don't try it. Or if you do try it, call me and I'll share our grant, because it was just very sad that we haven't been able to kind of test this in a real world clinical sample as a chronic treatment, right? More of our current thinking is around doing stress reactivity paradigms or understanding how oxytocin impacts things like craving in the lab differentially by HIV status. We also looked at alterations in reactome pathways, so similar to our pathway analysis from the mRNA gene expression work. We saw alterations relevant to immune function and, interestingly, neural function. So like dopamine release, neurotransmitter release, there was a recovery kind of signal that we were seeing, that as people were getting time away from stimulants like methamphetamine, that they were able to kind of regain some function, if you will, of these systems that could be impacted by it. So this was I think, really fun and somewhat wacky study. It made it into brain behavior and immunity, which we were very excited about. There was no change in any of the soluble markers we measured, right? And so in the paper, I said, this is great, and it highlights the benefits of epigenomic work. And it got reviewed by, I think it was like American Journal of Psychiatry, and they were like, well, yeah, maybe it's just like type 3 error. So, you have to be careful when you like present negative findings. I don't know, it's just really hard, right? Because I thought, gosh, I feel like we're holding this back and we should put it all together. But sometimes it's just easier to tell a story that doesn't undermine you. Good. And so we have 2 trials underway. One is mHealth application. So we've taken the positive affect skills and written a bunch of scripts, done a bunch of videos with someone who used to work with Judy, Alex Brusette, as a kind of talent doing them. We also connected with adult film star Boomer Banks, who's living with HIV and is in recovery from meth use. So he did a lot of cameo videos in this to make it interesting for guys. There's a lot of self-monitoring of adherence and mood in the application. And so we're testing this. We randomized 286 sexual minority men with HIV that screened positive for stimulant use disorder, so cocaine or meth. My recent thinking on this is that when we focus on meth alone, it just slants wider than cocaine/crack. And so if we're more inclusive of stimulant use disorders, I think we get a more diverse sample. So these guys were recruited from across the US. About half of them are racial or ethnic minority men. So we're actively following. They send in dried blood spots to measure viral load. So that is our primary outcome, a detectable viral load. And the cutoff for that is 300 copies because it's on dried blood spots. So news to come on that, I hope. And then the other one that we're doing with Mallory and also Laura, who I think was on the call or maybe, is ongoing. It's a two-site trial in Florida and California. And so we're doing telehealth 5 positive affect sessions. This time we took the contingency management that everyone receives and we made it about adherence. So guys log into an eMocha app and take videos of themselves taking their medications, and they get small incentives that escalate for doing that. Our primary outcome is HIV acquisition risk. So it's a low tenoffuran diphosphate level and reporting condomless anal sex. Coincidentally, everyone is on daily oral PrEP. We exclude event-based PrEP. We also excluded injectable PrEP given when this trial started. But of course, we're monitoring changes in people's PrEP regimens as they go along. 216 people have been randomized to date, mostly in California. Recruiting meth users in Miami is kind of fun. We finally found a peer individual that can navigate the networks, and we made a lot of inroads. But our target enrollment is 240 by March. So I think it will hit it. And then we're taking a bunch of next steps, but I don't want to keep you all longer. Cohort work, new RCT, funded with Christina Mead, and hopefully another NIDA cohort. And that's
And
it.
Adam, do you have a few minutes to answer questions from folks?
Yeah, totally.
Great. Yeah, I'm aware of the time. So if folks need to hop off, feel free to do that. But if you are interested in staying for a few more minutes and ask questions for Adam, please go ahead and unmute and yeah, connect with Adam now.
And you all know where to find me too. I'm happy to chat. We have lots of data, a lot of mRNA seq data to look at various, kind of pathways. The main one that we're interested in is the CTRA, the conserved transcriptional response to adversity. It was validated by Steve Cole at UCLA. So we always have data if folks want to look at it.
Adam, that was amazing. I'll ask maybe a silly question that you can disregard quickly, but I was just curious while you were talking about the HIV latency reversal possibility, like related to methamphetamine or stimulants. Have people explored that with regards to cure work, like whether people actively using stimulants may achieve cure differently? And I understand maybe challenges there, but curious.
Yeah. Silke Lee had like an R61, R33 on this topic, and we were going to give people oral methylphenidate to see, does that stimulant induce activation of the reservoir? I don't know where that study ended though. It's hard to do these trials, right? And there's a lot of noise and a lot of ethical issues, obviously, to give people stimulants. But that's the kind of experimental data that we would need. And kind of fundamentally, you ask yourself, like, is the cure to HIV in someone that uses meth? And that would be, I think, so beautiful and destigmatizing. But it could wake and kill the virus for sure.
Thank you. That was an amazing talk.
Yeah. Thank you.
Adam, is it okay if I share your email on the chat for folks who want to reach out? Great.
Yeah.
Other questions for Adam? I also saw there were a few questions on the chat. I don't know.
I have a question.
Oh, go ahead, Susan.
Hi, Adam.
Hello.
Hey.
Hi. So have you, in your positive affect stuff with Artemis, I know it's all one-on-one approach that you talk with, have sessions one at a time. Have you tried anything with group level approach?
No, but this came up with Thomas's ideas as well.
Yeah.
We haven't, but I think that's much more scalable, right?
Yeah.
And there's the theoretical kind of underpinning of revised stress and coping theory is that positive affect generates, and the broaden and build is related to this, but it builds social resources, cognitive resources. So if it was done in a group, it could potentiate, greater engagement with your peers, which would be fine in general if people aren't actively using. I've always been worried about doing group work with active users where there's not like a commitment to sobriety or, like sometimes peer support and engagement is a bad thing for people that use drugs. So you just have to think about like iatrogenic effects in that regard. But definitely with, young MSM or, folks that aren't actively using as a preventive kind of intervention, that would be really exciting.
Yeah, but the issue about introducing guys to new social networks of users is concern.
Yeah, and it always is. I mean, in 12-step groups we see this, in treatment we see this. It's not like we can completely insulate the world or the treatment environment, but there are pros and cons to any modality.
Yeah. Thank you. I'll be contacting you shortly.
Right on.
Just a quick question, the CBT, adding CBT to CM or whatever it was, what was the name of the author on that?
Oh, I'll
You said
find
there
that
was,
one. I'll
okay.
find that one.
I was very
With
curious.
CM doing better than CBT for durability.
Yeah, durability, that's right. That's what, yeah.
Yeah. I'll find it because I was like,
Thanks.
dang, someone finally did it. Because every time I would talk about this trial, I would say, this is what we think, but there's not data to support this, that CM rapidly relapse and after the incentives. Yeah, it does really
We
open a window of people for people. Like it gets them time away from drug, it gets them a little clean headspace. And I think that can make a lot of difference.
we had a 12-month alcohol effect after 6 months. 6 months later, still had our effect at 12 months. So
That's
yeah,
awesome.
yeah. So we're starting to, I think the CM, we're starting to think maybe it's not, so short term.
Yeah, right. But it's hard again, it's fighting against the dominant narrative that people have in their head. And that's going to be an uphill battle. Well, thanks everybody.
Thank you so much,
Thanks,
Adam.
Adam.
That
Thanks,
was
Adam. So
wonderful.
awesome.
Always a pleasure having you and seeing you.
Same. See you all soon, I hope.
Please take
Bye.
care, everybody.